start too early. Checking my watch. I know it's been like a week since we last met. So I hope all of you enjoyed Nobel and maybe the time off that Nobel afforded you. I saw all the speakers and I thought they were fantastic. Does anybody have any favorites? Or maybe like an event you saw instead of a speaker? Yeah, Jonathan Birch's lecture. Yeah, this was pretty cool. Yeah, I like Julie, I think it's Julie Lesnick. She's an anthropologist. Interesting. Yeah.
I really like the bumblebee with the ball.
Yes, the bumblebees with the balls, yes. I thought they were all really fantastic speakers this year. But I could be biased because I am already insect.
Did you eat the bugs?
I did eat bugs. I saw Max at the bug reception too. I don't know, did anybody else go to the bug reception?
I did.
Yeah. It was good, yeah.
I just told them I was hoping they weren't serving any leftovers during
Yes,
thank you.
family weekend on campus, so there's a lot going on. Personally, I will say I did have a pencil class on Monday. My dad did have a stroke. He is on the road to recovery. I may have to cancel classes in the future. I will give you as much notice as I possibly can if that is the case. All right, but he's doing good. We're all super happy and thankful that he caught it in time. But I did want to let you know that in the aspect of just being transparent with you all. All right, so exams. Yes, they are graded. No, they are not getting passed back quite yet. The reason being is there was a sickness on campus last week, right? So I still have another student that has yet to take it, so I can't give it back yet. But if you come to my office after class around 1:45, I can give you your exam, all right? It will be good to be handed out at that point, all right? If you can't come to my office after class, I will make sure to pass all the rest of them back on Monday. All right. Let me talk about the exam before I take questions. All right. So the class average was a 76%, which is fine. That's kind of what I would expect for the first exam, especially first science exam for many of you. Keep in mind that exams are not the end all for your grade here, right? You have labs. You have those scientific literacy assignments, which are more or less guaranteed 10 points. You have the content checks, which I drop your 3 lowest scores. And you have those complete and incomplete assignments, plus 3 more exams this semester, all right? So you know what to expect from an exam now. You kind of know my format and the breadth of what I will give my exam on. So that's the good news moving forward. All right. Now, I did post your grades on Moodle, so you can look at your grade if you are curious. I like data. All right. So I took the data from all of the questions and compiled them because I feel like if more than 50% of you got a question wrong, right, it was either I didn't teach it right or the question was just asked very poorly. All right. And so that red line is that 50% there. So there are 2 questions above that line, right? And I'm not going to tell you which questions they are right now because, yes, we still have another person who needs to take the exam. I will send out an email with this information after class, right? There is one question that I am automatically giving you that point back if you got it wrong, right? If you got it correct, awesome, great job. You got it wrong, you get the point back. So essentially this exam was out of like 44 points instead of 45 points, right? Yes. Yes, that has already been reflected on the board, right? This last question here, it was a multi-part question, which is why it's a little bit higher than 50%. because I took if you got any part of it wrong, right? And then I have these 3 with the green stars that were just ever below 50%. They're like 46%, right? So I rounded it up, right? So for 4 of these questions, I'm going to give you the opportunity to earn points back, right? So when you collect your exam from me, right, and this will be in an email as well, I want you to correct the answer. I want you to write your rationale for why you corrected it, and I want you to turn that back into me. And I will give you up to half points back for all of those questions, right? Now, keep in mind, even 1 point on these exams is like an extra 2% boost in your score, right? So again, I'm going to send out an email with all the specifics on that, and if you have any questions, please let me know. All right, before I move on to content, any questions about the exam that I didn't address? All right, again, it's your first exam in my class. You've never had me as a professor. You've never had my exam as a teaching style. I think the results were absolutely fantastic. All right, so nice job, all of you. Don't fret if it wasn't a grade that you necessarily wanted. You can talk with me if you'd like. Come to office hours, send me an email, whatever you would like to do. All right, so we're going to jump into the next unit. All right, and the next unit is all about cellular respiration and photosynthesis. Right? So this unit is a lot about pathways and processes. Right? And so we're going to start by talking about cellular respiration, which you touched on a little bit in lab a couple weeks ago. Right? We're going to catch up with the lab material now, and next week you'll have a lab on fermentation, and we'll be talking about fermentation next Wednesday. Right? So we should start to be in sync with lab again. But we're talking about metabolism. Right? Metabolism is all of those chemical reactions inside of a cell that are life-sustaining. There are a lot of them, right? This is a giant map. It's almost like a piece of art the way it's laid out here of all the different chemical pathways that we might consider being a part of metabolism, right? Another way we can think of metabolism is converting the food that we are ingesting into a currency of energy that our cells can use. So yes, ATP will be a big molecule that we will be talking about today and for the next week and 2 weeks here. So if we talk about these metabolic pathways, there's a lot of them, right? We can generally split them into 2 different kinds. The first would be a catabolic pathway, right? So this is a pathway that breaks down cellular components, right? We take a molecule, we break it down into smaller parts, right? These reactions are generally exergonic, meaning they release energy, or they happen spontaneously, right? So think back to when we talked about enzymes and chemical reactions, all right? So they happen spontaneously. You're breaking them down, they're releasing energy. The other type of pathway is an anabolic pathway, right? And this is exactly the opposite. Instead of breaking something down, we're building something up, right? These are not spontaneous. They are endergonic reactions, right? They require an energy input in order for them to happen, right? So I really like this example of this image up top here where we see these molecules being broken down, releasing energy for catabolism. And then we have these smaller units being built up into larger molecules with the input of energy in anabolism, right? Now it's really important to note that a lot of these reactions, most of these reactions, are catalyzed by enzymes, right? And so if we look at this generalized reaction here, right, we are going through some process. It doesn't matter what that process is, but each step is catalyzed by an enzyme, right? So there's a bunch of enzymes, hundreds of enzymes, when we talk about metabolic pathways. Now I want to go on just a little bit more detail into these 2 pathways, right? So if we talk about these anabolic pathways, we can say that they are biosynthetic reactions, right? So Biosynthetic. The word synthetic coming from synthesis. So we're building something, we're synthesizing something. And bio meaning biological, right? So these biosynthetic reactions, right? Oftentimes these types of reactions will be used to make large macromolecules. If you remember back to when we talked about carbohydrates, we talked about like starches and glycogen, right? Those are large polymers built of sugar molecules, right? To build those molecules, you would use an anabolic reaction, right? Or that we might be building molecules that aren't quite as big, right? But they're still larger than the smaller components that make them up, that we just simply can't get from food, right? So we're building other molecules from those food substances to make other molecules that we can't directly get from food. And again, they require energy to drive these reactions. Often this energy is in the form of ATP, which we have talked about, but also NADH, right? Keep that molecule in mind. We will talk about it in a few slides. Right? And again, the opposite of that is that catabolic reactions, or breaking down those molecules. Often this is used for like recycling of those building blocks. So I really like this image here. You see that we're taking in food. Food is made up of these polymers here that are either proteins, carbohydrates, fats, right? So if we say, look at the proteins here, we need to break them down into their building blocks, which would be amino acids. And then once they're broken down, those amino acids can go and build other proteins or be used in other ways. Right? So we're breaking down those big molecules into their component parts so we can recycle them. But we can also break down molecules for their energy. Right? So think ATP, hydrolysis of ATP. Right? We know that's a spontaneous reaction. We know in the bonds of ATP it holds energy. When we break it apart, it releases that energy. Right? And so this is another big component of these catabolic reactions, is breaking apart those molecules so we can use the energy. All right, let's step back and think about what we just learned and answer this question here. So for each of these reactions, I want you to identify the type of reaction. Is it catabolic or anabolic? All right, I'll give you about 2 minutes here. And we'll go from there. Let me know if you have questions. Work with your neighbor, talk to each other. How's it going? Good. I was worried when I had I get the sense that we've all answered this question already, and maybe 2 minutes was a lot longer than we needed. This first example here, who wants to answer what type of reaction it is? Yeah, it's catabolic. Yes, we are breaking something down into its smaller components. We see we have a large cellulose molecule being broken down into individual glucose molecules. And this other one, anabolism or anabolic, yes. Yes. We are building up a molecule from these two components into a larger component. Does anybody recognize what molecule this is? Nucleotides building up a DNA strand. Yeah. All right. So I wanted to do a quick review of ATP, and I think this video does a really good job of just like synthesizing what we've learned so far. And plus it's a visual, right? I love visuals.
That will be used to facilitate such reactions is called adenosine triphosphate, or ATP. ATP is essentially just an RNA nucleotide. Notice the adenine base, the ribose sugar, and the phosphate groups. The only difference is that in a nucleic acid like RNA, each nucleotide has one phosphate, but here there are three, and it is these phosphate groups that give ATP its energy-storing properties. There is an abundance of negative charge on these phosphates, and we know that like charges repel one another. So there is a lot of potential energy stored in this part of ATP, kind of like a compressed spring. Just the way a compressed spring wants to expand, releasing the stored potential energy and converting it into kinetic energy, these phosphates want to come apart. So when ATP undergoes hydrolysis and transfers one phosphate group to something else during an enzymatic reaction, thus becoming adenosine diphosphate, or ADP, it releases some of that potential energy, which can then be converted into the energy needed to promote a reaction, or pump ions across a membrane against the concentration gradient, or any number of other things. ADP can then be phosphorylated to become ATP again. So this back and forth between the two forms is the method that nature has discovered to provide energy for cellular processes.
All right, so hopefully that rings a bell. We talked about ATP hydrolysis when we talked about enzymes here. I think that was a really good review of what we talked about. Well, and we don't need to listen to it again. There we go. We also have this other molecule that I said, keep in mind, right? It's NADH, right? So this is another molecule that helps provide energy, but in a different way from ATP. Right? We would call NADH an energy intermediate. And what this molecule does is it can accept and donate electrons. Right? And that acceptance and donating electrons can transfer energy to other molecules. Right? So we can it releases a lot of energy when it loses electrons, and that can be really helpful in making ATP. Right? We'll see this in the final step of cellular respiration when we talk about it on Monday. When it donates those electrons during synthesis reactions, so we're building up molecules, it can help energize that reaction, give energy to that reaction. All right? So NADH is another big molecule we will be talking about here. But in order to really understand it, we have to understand redox reactions. So, oxidation, reduction reactions. So maybe you remember this from chemistry, maybe you have learned about chemistry this semester, or maybe you have no idea what this is, right? This is essentially a reaction involving an electron transfer, right? I'm not going to get into the nitty-gritty here. I want you to learn the big concept here, right? So we have oxidation, which is the removal of electrons. And we have reduction, which is the addition of electrons. And yes, I know the wording does not make sense, right? Reduction, we're adding something instead of reducing something, right? It's kind of counterintuitive. But you can think of it as a reducing of charge, right? If we're adding these negative electrons, we are reducing the charge by adding more of a negative charge, right? Maybe you've heard the term oil rig when learning this, right? So oxidation is loss of electrons, and reduction is gain of electrons. So that can be a helpful way to remember this. if we look at a very, very, very simplified reaction here, right? We have molecule A, molecule B, right? And in this reaction, this electron is simply going from the A molecule to the B molecule, right? So if we look at the products here, we can see that A has been oxidized because it has lost an electron, and B has been reduced because an electron has been added, right? Now this is really simple, right? It gets a little bit more complicated, as everything does, right? So here we have the, yes, it's a lot. I'll walk us through this. We have the general reaction for cellular respiration up top, right? And when we have these redox reactions, it's often not just an electron moving, but it's an electron moving with a hydrogen ion, right? And so this reaction is really what's illustrating how these hydrogen ions accompany those electrons. So if we look at this glucose, for example, right, and look at the CO2, we can say that it has lost hydrogen atoms, right, which have those electrons with it, right? So it has become oxidized. Same with this oxygen, right? It has gained hydrogen atoms, which have the electrons with it, so it has become reduced, right? So when you're looking at these equations, yes, it's the electrons moving, but oftentimes you're not going to see the electrons written in the equations, and you'll have to follow the hydrogens instead, right? It has to do with balancing of charges. For moving a negatively charged electron, we want something positive with it, and it just so happens a hydrogen ion is positively charged. So they often move together. Now I put this other image down below here, all right? I don't expect you to map out electron movement in this way, but it could be helpful to understand oxidation and reduction, all right? This is the same exact equation as up on top here, but it has mapped those electrons out and how they move. And we can see electronegativity can also play a role in whether something is being oxidized or reduced, right? So oxygen can play a huge role here. It's really electronegative. It hogs those electrons, all right? So we could say that the carbon in this case is being oxidized, not because it's totally lost an electron, but because now oxygen is pulling it away. Oxygen is hogging those electrons, right? So electronegativity, it doesn't go away when we move on to the next unit. It's still here. It still plays a role. But I really want you to focus on this top piece here. Don't get bogged down in the details, all right? It's not a chemistry class or a biology class. So focus on big picture here. Oop. All right, so back to NADH. Right? NADH can go through these redox reactions, right? And that's how it transfers energy to other molecules. So if we look at this molecule over here, right, it is NAD+, right? We see that if we add on 2 electrons and a hydrogen, right, so we're reducing this molecule, we get NADH, right? And notice the change that has happened here is on this top hydrogen, we go from that 1 hydrogen to adding in a second hydrogen, right? One of the electrons goes along with that hydrogen. That other electron will add to that positively charged nitrogen, and we now see this nitrogen over here no longer has a charge, right? So NADH, it accepts and donates electrons, and this is specifically how it does so. And it can bounce back between being NAD+ and NADH, depending on if it has gained those electrons or lost those electrons. All right. Again, I have this nice video that I don't know why it wouldn't play within my slides, so I have to exit out and go to this video. But it does a nice job of reviewing NADH. Let me get in a full screen here.
NAD+ is a carrier molecule that traps electrons and protons and transfers them to where they're needed in the cell. And that's the best way of describing it. State that it's a carrier molecule, state that it traps electrons and protons, and finally that it transfers those electrons and protons to where they're needed in the cell. It's really important that you know how NAD+ traps the electrons and protons, and where indeed are these electrons and protons coming from? Well, firstly, the electrons and the protons are coming from hydrogen atoms, pairs of hydrogen atoms, and so whenever we consider NAD+ in cellular respiration, we always consider electrons and protons coming from pairs of hydrogen atoms. So the details of this reaction are NAD+ will trap 2 electrons and 1 proton, remember they're coming from hydrogen atoms, and so is converted or is reduced to NADH. And when you compare NAD+ with NADH, we consider NAD+ to be a low energy molecule and NADH to be high energy. To make our equation perfect, we have to account for that one other proton. Remember, we're talking about pairs of hydrogen atoms, 2 electrons and 2 protons. So there's one other proton that needs to be added into the equation. So let's sum up. NAD+.
All right, I'm going to pause it there. Maybe.
Molecule, and it traps electrons and protons and transfers these to where they're needed in the cell. The electrons.
All right, there we go. So I think, again, that video does a really nice job of kind of synthesizing that big picture idea in a way that's a little bit easier to understand. So let me bring up my slideshow again here. I have another group question for you all. All right, I want you to just review how exactly does each molecule provide energy for that cell, right? And then as we're thinking about this might be more of a review question from last unit, but what organelle is going to play an important role in cellular respiration? Well, we can go ahead and move on since I don't hear any discussion. So, how does ATP provide energy? Where is the energy stored? Yeah, in the bonds in those phosphate groups, yeah. Right, so it holds those energy in the covalent bonds. And when we break off a phosphate group, we're releasing some energy that, as we learned, can be coupled with reactions to help them spontaneously go. What about NADH? Where is the energy held there? It's not in a bond. has to deal with hydrogen. Hydrogen ions and electrons, right? Yes, the big party there. So an NADH, it's the removal and acceptance of electrons that transfers energy, right? So they're both energy carriers, but they carry that energy in different manners. Now, what organelle is going to play an important role in cellular respiration? Mitochondria, yes, mitochondria, the powerhouse of the cell because it produces so much ATP, but also NADH. All right, any questions? All right, we're talking about pathways, right? So we have to talk about how these pathways might be regulated, right? So they're not just completely having these reactions going all the time. There's some regulation involved. So one of the ways we can regulate these pathways are through gene regulation, right? Turning genes on or off, right? So a good example of this might be if there's a sugar molecule that we need to break down, right? Maybe that gene is turned on in the presence of sugars, but if there's no sugars present, maybe that gene is turned off, right? So we're not constantly producing the enzymes and everything needed for this pathway all the time. Another one would be like cell signaling, right? So cells can talk to each other, signal to each other. And a lot of times those pathways in which they signal to each other can activate what we call protein kinases, which are an enzyme that are involved in cellular respiration or other types of reactions. All right, so that cell signaling, cells can talk to one another and say, hey, it's time to turn on that enzyme, we need it now. Another one is this biochemical regulation, which would be feedback inhibition. All right, and so this is where in the process of a certain pathway, the products are actually going to regulate whether that pathway is going to continue or stop. All right, and so if we look a little bit closer at this feedback inhibition, we know living organisms, we need to maintain homeostasis, right? So we need to regulate the amount of substances in our cells. Right? So too much of a good thing can end up being a bad thing. So we need to have a way to regulate this. And feedback inhibition is one that we will see fairly often. And this is where the final product of a pathway will actually build to a certain concentration, where then that product is going to go back and inhibit that same product from being produced. So if we look at this abstract pathway here, right, we get to our final product. That final product, once it reaches a final concentration, will bind to that enzyme that began the whole pathway. And that acts as an inhibitor, right? It actually changes that active site of the enzyme so that it can no longer bind to the substrate and the pathway stops because we have enough of what we need already. Now, as that concentration decreases, right, this product will unbind and we can go through the pathway again, right? But we see many different types of regulation in this manner, this feedback inhibition. So much so, Kat, I want you to think of examples, all right? So let's take a few minutes here. I want you to use Google, use the internet, and figure out what pathways need to be maintained. We don't talk about animals enough in physiology, so I thought this would be a good chance for us to dive into a bit. I don't know what I'm talking about. Yeah, everybody has an example. All right, what are some examples of maintaining homeostasis? Yeah, blood pressure, sweating, maintaining that internal temperature. Yeah, osmoregulation, regulation of water. What else? Yeah, regulating pH. Different pHs in different areas of your body. Yeah, heart rate when you're exercising. Like glucose levels, body temperature. There are so many, right? Now, not all of them use feedback inhibition to regulate their homeostasis, but a lot of them do. So it's a pretty important mechanism. All right, so let's move on to actually talk about cellular respiration. Right? And if we get through all of our slides, it's great, and you'll learn the first step of cellular respiration. But we're going to start on it and then finish this up on Monday. So, cellular respiration, right? That is a process by which living cells obtain energy from organic molecules, right? And I have this nice diagram up here showing mitochondria, where cellular respiration mostly takes place, but also photosynthesis, because oftentimes they kind of go hand in hand, right? So some of the major products of cellular respiration would be water and CO2, in addition to all that chemical energy it produces, right? Chloroplasts, or plants that might photosynthesize, other organisms that photosynthesize, they use CO2 and water, as well as energy from the sun, to then produce glucose and oxygen, which then we use in cellular respiration, and the cycle continues, right? So they go nicely hand in hand, which is why we talk about cellular respiration and photosynthesis in the same unit. Now, cellular respiration is not limited to animals. Plants do this too, right? We often think of plants as only photosynthesizing, right? But they need to use cellular respiration, right? Think about a seed that is germinating, right? It's currently germinating. It does not have any leaves yet. It cannot photosynthesize. So where is it getting its energy from? Well, it's using cellular respiration, right? A lot of cells in the roots of plants, because they're not exposed to sunlight, will use cellular respiration. Plants at night, when there's no sun, can use cellular respiration. A what? Yeah. So like a tulip bulb, there's a lot of energy stored in different compounds that they will then use cellular respiration to break down to produce those leaves so that they can then photosynthesize. Yeah, so plants use cellular respiration. I really wanted to hone in on that because oftentimes that is forgotten about. All right, so if we look at an overview of cellular respiration, right, the primary aim is to make energy. Energy in the form of ATP and NADH, right? We're really going to start by talking about aerobic respiration, all right? So this is the type of respiration that those cockroaches used in lab, last week? Yeah, last week. I want to say 2 weeks ago, but it's not been that long. Last week, right, we looked at aerobic respiration in lab, and aerobic respiration uses oxygen, all right? And if you had me in lab, you probably saw this diagram, right? If we look at respiration, there is a pathway that uses no oxygen, right? And eventually we'll go into fermentation. We'll talk about that on Wednesday. But it does produce some energy, some ATP, but very low amounts. Aerobic respiration, which is what we're going to talk about today and on Monday, uses oxygen and produces a lot more ATP, up to like 36 ATP molecules, compared to just the 2 in the absence of oxygen. Right? So aerobic respiration used by organisms that need a lot of energy, right? So like ourselves, right? Most of the respiration we do is aerobic respiration, right? We can do anaerobic respiration. We can go through fermentation. You might see this sometimes when you're exercising in your muscle cells. They just can't get enough oxygen in, and we'll go through a fermentation stage. But we can't sustain ourselves on just fermentation alone. Now, in cellular respiration, a lot of times the organic molecule broken down is glucose, right? And we're going to use glucose as our example when we talk about cellular respiration, but know that other organic molecules can be used too, all right? Glucose is kind of like the model molecule when talking about cellular respiration. All right, so if we talk about glucose metabolism, there are 4 metabolic pathways that we are going to go over, right? There is a lot of detail here, right? If I want you to know a certain molecule, I will often bold it, but I certainly do not need you to memorize all of the molecules involved in cellular respiration, all right? So there is a lot of detail. Focus on big picture and understanding how the process works. So the 4 pathways would be glycolysis, breaking down of pyruvate, the citric acid cycle, also called the Krebs cycle, okay, and then oxidative phosphorylation. Okay. Again, this ultimate goal of all of these steps is to make energy, ATP and NADH primarily. All right, so if we look at those 4 pathways splayed out here, we see that a lot of it happens in the mitochondria, right? But this first step, glycolysis, which we'll talk about today, is actually happening in the cytosol, right? So it's not happening in the mitochondria, but rather that fluid surrounding the mitochondria, surrounding all of the organelles in our cells, right? And then the next 3 steps will happen on the inside of that mitochondria. So if we first look at glycolysis, right, that's step 1, right? This is where we take our glucose molecule and start to break it down. Now, glycolysis can occur with or without oxygen, right? If we're going through aerobic or anaerobic respiration, glycolysis can happen in both ways, right? Now, if it's anaerobic, we don't have oxygen, cellular respiration will end after this step, right? But with oxygen, we can keep on going. So in glycolysis, we can break down all of the different steps into 3 main stages, So in total, there's about 10 steps, right? These 10 steps, this is highly conserved in evolution. Most organisms will have these same identical 10 steps in them, right? But we can break them down into an energy investment phase, into a cleavage phase, or we break something apart, and into an energy liberation phase, where we actually get a little bit of energy out of it. So if we look at this first phase, right, I know, again, this is a lot of steps, a lot of details. Focus on what I have bolded, right? This first phase is energy investment, right? Called energy investment because we actually need to use some ATP in the first steps here, right? So we are actually using ATP for its energy, right? So these two ATP will be hydrolyzed, and it's going to turn this glucose molecule into this intermediate molecule called fructose 1,6-biphosphate, right? It's called 1,6-biphosphate because we have phosphate groups on carbons 1 and carbon 6, right? So that's that first step. Big picture here, right? We are using ATP. Now, in that second phase, we call it the cleavage phase, because now we are breaking apart this intermediate molecule into two glyceraldehyde 3-phosphates. And yes, that is bolded because I think it's an important molecule, and it's a molecule we'll see again in photosynthesis, right? And so in this phase, we're taking apart that fructose 1,6-biphosphate. We're splitting it into two. So we see this fructose 1,6-biphosphate has 6 carbons. We are now breaking it down into two 3-carbon molecules called glyceraldehyde 3-phosphate, often abbreviated to G3P, right? And we are now ready for our next phase, right? So again, big picture, breaking it down into two G3P molecules. That last phase of glycolysis, right? This is where we actually get some energy, right? So we're taking these glyceraldehyde 3-phosphate molecules. They are each undergoing the next few steps independently, right? And they are broken down into two pyruvate molecules, right? Through a series of like 1, 2, 3, 4, 5 different reactions here. Now, throughout those series of reactions, we are getting ATP, right? It is producing ATP, right? 2 ATP molecules per G3P molecule that's being used, right? And we're getting 2 molecules of pyruvate. Right? Now, not only are we producing ATP, but we're also producing some NADH, right? So if we look and think big picture here, our net yield of ATP, and these are just all the steps that I had previously listed on the last 3 slides, and I just combined them into one slide for you here. All these steps are yielding 2 molecules of ATP. So we had to use 2 molecules in the first step, and we produced 4 molecules in that last stage, which gives us a net of 2 ATP molecules that have been produced. Yes. It is the first step in fermentation. Yes, it's the first step in fermentation. It's also the first step in cellular respiration. Yeah. Because then we have to regenerate those pyruvate molecules. So those pyruvate molecules in fermentation will then go on to a few other steps. It's not going to yield any more energy. So yes, you're correct. Fermentation only yields 2 ATP. But there's other steps that happen afterwards that don't produce energy but are still important. Yeah. Any other questions here? All right. I think I'm going to end at this slide. I know there's a couple more slides after this. We will finish this up on Monday. Before you all leave, though, I'm just going to skip ahead to the homework for Monday. All right. I'm not assigning you any homework for Monday, but I did put the content check up, contact check number 3 that is due on Wednesday up. That Nobel assignment, it is worth 2 points on your next exam. Right? If you did not see any lectures, they are recorded. You still have some time to get that assignment into me by tonight. Right, and I will be in my office in roughly 15 minutes if you want to collect your exam. Yes.
Brandon makes being blind is easy. What's that? Brandon makes being blind is easy. At least when it comes to Bible 101. Things are insane. It's crazy how we do it. Like, you all have the slides and like everything in here. Okay, so how does it work though?
There's actually a button that if you present on Google Slides, I had to learn this, but there's a button in the corner where you can close, open and close closed captioning.
Wow, that's so
I know. So the technology maybe exists now to make this easy.
That's so cool. Can I like
Oh, yes. Yes, I know. Several people missed that last question. So,
but I
yeah.